Hello everyone. Let's start chapter 12, and that is about muscle. As you know, we have three different types of muscles: skeletal muscle, cardiac muscle, and smooth muscle. In this chapter, we're going to focus on the skeletal muscle and smooth muscle, and we're going to talk about cardiac muscle in the next chapter. I'm sure you all remember that we mentioned in the lab that the skeletal muscle is composed of a bunch of cells, and these cells are striated. Each cell, in this picture you can see three cells, one here, one here, and one here. Each cell is called a muscle fiber, and the cell membrane of this muscle cell or muscle fiber is called sarcolemma. Let's say this is the muscle, and here shows a bunch of muscle cell. Each one of this one is one cell. Again, it's called also muscle fiber. To contract these muscle cells, each cell one by one must be innervated. For instance, if you cut this one here, this cell is going to become paralyzed. If you cut this one here, this one becomes paralyzed. Although it doesn't show for the rest of the cells, but you have to realize that every cell inside the muscle is being innervated one by one. A neuron is going to come, and at the end of the neuron, you have some vesicles. This vesicle is full of neurotransmitter and neurotransmit acetylcholine. Acetylcholine will be released and it's going to sit on this receptor. The receptor here, in the case of skeletal muscle, is nicotinic receptor. And as a result, your muscle cell will contract. I'll show you how this contraction happens. But before we do that, let's take a look at the structure of the muscle. When you look at one muscle cell, you can see inside the muscle cell, you have an organelle that's called myofibril. Myofibril is an organelle that can contract. As a result, your whole muscle can be contracted. When you take this myofibril out and you look at a structure of the myofibril, you notice that your myofibril is composed of some other structures called the myofilaments. So this is my myofibril. Each of these one is one myofibril. And as you can see, myofibril is composed of myofilaments. Myofilaments itself is composed of two components: thick and thin filaments. This is my thin filaments, and this is my thick filament. Thick filament is composed of a protein is called myosin. As you can see, one molecule of myosin is composed of one tail and two heads. The head of the myosin has ATPase activity. That means the head of the myosin can break down ATP. The thin filament is composed of three different types of proteins. These beads are called actin. This rope is called tropomyosin, and this structure here is called troponin. Troponin is abbreviated by TN. Troponin is composed of three pieces: TNI, TNC, and TNT. This picture shows thin filament. These are actin, this is tropomyosin, and this is troponin. Let's say this one is your TNT. It's called TNT because it attaches to tropomyosin. Then you have TNC. Let's say this one is TNC because it binds to 4-calcium. And let's say this is TNI. I stands for inhibition. I'm going to explain the function of TNI in just a few minutes. In this picture, you can see this is your thick filament. Thousands of thick filaments. The tail is going to wrap around each other and the head is going to stick out. These are the actin, these beads. They are composed of some area. It's called active site. The head of the myosin always wants to attach to active site of the actin. However, when the muscle is relaxed, tropomyosin, this rope, is going to cover the active sites. Therefore, the head of the myosin cannot attach to the actin because active sites is being covered by this tropomyosin. You have to understand that TNI, this member of troponin, also is going to help tropomyosin to cover the active sites and prevents the head of the myosin to attach to actin. So TNI and tropomyosin, they share the same function. If you look at this picture, you can see how the thick filament and thin filament are arranged. As you can see, we have thin filament, thick filament, thin filament, thick filament. They are interdigitated. These are proteins, and they have to be anchored to something. They cannot just stand in the air by itself. The thin filament, as you can see, is attached to another protein. It's called Z-disc. In this side, you can see thin filament is attached to another Z-disc. The space between one Z-disc to another Z-disc is called sarcomere. And what is the Z-disc? This is another protein. I want you to think about this one like a scaffold. When you create a scaffold, Obviously, there are a bunch of middle tubes. They cannot stand alone. However, when you link them to each other, then it's going to create a stable structure. The same is true about this one. Thin filament by itself is not going to stand alone. But when it's attached to Z-disc, then it's going to create a stable structure. In the middle of this thick filament, we have another protein. It's called M-line. Each of this area I'm showing you, this one, this one, this one, these are M-line. M-line also is a protein that creates stability for myosin. Let's learn some terminology here. The space that only contains thick filament and doesn't have thin filament is called H-band. So from here to here is H-band. This area, which is composed of thick filament, is called A-band. So from here to here is A-band. The part that doesn't have thick filament is only thin filament, just like this picture. Again, this is the thick filament, this is thin filament, this is a Z-disc, this again another thin filament, and this thick filament. From here to here is called I-band because it does not have thick filament. So we have H-band, we have A-band, we have I-band, and I want to know the term sarcomere. Remember, M-line is right in the middle of this thick band. Now, besides the protein that we mentioned, myosin and actin and troponin, tropomyosin, M-line, Z-disc, these are all protein. Besides those proteins, there are other proteins that is worth to be mentioned. One of them is dystrophin. Dystrophin is a protein that is going to attach the whole structure that we mentioned into the cell membrane. I know it seems like very complicated, but I want you to take a look at this one. This is my thin filament. And thin filament through the dystrophin is going to attach to sarcolemma, which is cell membrane. In some diseases, like Duchenne disease, you will see dystrophin will be missing and is impaired. The next protein that I want you to know is called titin. Titin is going to attach your thick filament to Z-disc. Let's take a look. This is your titin, this green one. As you can see, it's going to attach your thick filament to the Z-disc. Titin is a protein with elasticity. Muscle is like a rubber. You can pull the muscle and make it stretched. The stretching ability of the muscle comes in part because of elastic fibers, and the one that I want you to know is called titin. The next protein that you should know is called nebulin. The nebulin also is associated with thin filament. In this picture, this blue one is nebulin. As you can see, nebulin is going to attach your thin filament to the Z-disc. And the last protein that I want you to know is called alpha-actinin. Alpha-actinin also is going to anchor the thin filament to the Z-disc. In this picture, you can see alpha-actinin. Although it's not a perfect picture, this line is supposed to be alpha-actinin that attaches to thin filaments. I put this picture here not to scare you, but I want to show you that what you saw in the previous picture is very schematic and very simplified. You don't need to memorize anything in this picture. I just put it here to give you an idea that we have many different types of proteins in the skeletal muscle cell. Now let's go to our schematic picture. As you can see here is the head of the myosin, which by the way is called also cross bridge. And here's my thin filament. Intuitively, the head of the myosin wants to attach to actin, but it's not going to happen because tropomyosin is right here and TNI prevents this attachment. What it takes for this head of the myosin to attach to actin? The answer is calcium. As soon as calcium is available, calcium is going to attach to TNC. By attachment of calcium to TNC, troponin, this complex, is going to change the shape. And remember that piece of this troponin, which is called TNT, is attached to tropomyosin. As a result, by changing the shape of troponin, this tropomyosin is going to move away and the head is going to attach to exposed active site. Remember, the only thing we need to attach the head of the myosin to the active site of the actin would be calcium. The amount of calcium inside the cell is pretty low. As a result, your muscle is not in contraction. However, as soon as the amount of calcium inside the cell goes up, then the contraction is going to happen. The word contraction simply means the head of the myosin is going to attach to actin. In a normal relaxed muscle, the amount of calcium inside the cell is 10 power minus 7. As soon as it's going to go to 10 power minus 6, then calcium is going to attach to troponin, the tropomyosin is going to go away, and the head of the myosin is going to attach to actin. I also want to talk about another component that we need for our discussion, and that is called T-tubule. In this picture, this is my cell membrane. It's called sarcolemma. These are my myofibril. This blue structure is endoplasmic reticulum. In the case of skeletal muscle, we call it sarcoplasmic reticulum. Sarcoplasmic reticulum in the case of skeletal muscle is very well developed and extensively covers your myofibril. There is an extension of the cell membrane that comes close to this sarcoplasmic reticulum, and that is called T-tube. T-tube is the extension of the sarcolemma inside the cell. T-tube is going to be wrapped by sarcoplasmic reticulum on each side. As you can see, sarcoplasmic reticulum is going to come close to T-tube and it's going to expand. This expansion is called terminal cisterna. So there's a terminal cisterna on each side of the T-tube. That is going to create a triad because now we have three structures: a terminal cisterna here, T-tube in the middle, and a terminal cisterna in this side of the T-tube. Inside the sarcoplasmic reticulum, we have lots of calcium. To release calcium, we have to bring action potential to this T-tube to release calcium. Let's see how we're going to release this calcium, because release of calcium is required to create contraction. Well, the answer is the action potential is going to come through this neuron and it's going to reach the end of the neuron. To the end of the neuron, we have vesicle full of neurotransmitter acetylcholine. When you release this acetylcholine, it's going to sit on the nicotinic receptor. Remember, nicotinic receptor is going to open up and sodium is going to come inside the cell. As a result, this cell becomes depolarized. As soon as this area becomes depolarized, this depolarization is going to open some other sodium channels. These are the sodium channels. And as a result, the sodium keep coming in. So as you can see, one is going to open the next one and the next one, and it's going to open the next one. And remember, the sarcolemma is going to penetrate inside the cell. It's called T-tube. As you can see, the wave of this depolarization is going to get to the T-tube. Remember, T-tube is close to the terminal cisterna. And remember, terminal cisterna is full of calcium. At the T-tube, we have another protein, another channel here. That this channel is called dihydropyridine, or DHP. So this is my T-tube. This is my sodium channel. The wave of depolarization is going to get inside and it's going to get to this purple channel, and this purple channel is called DHP, or dihydropyridine. DHP, or dihydropyridine, is a voltage-gated calcium channel. However, no calcium is going to pass through this channel. What really happens is this channel is attached to another channel that you find on the terminal cisterna, this yellow one. Remember, this is terminal cisterna. And this yellow one, this yellow channel, is called ryanodine. Ryanodine is physically attached to the DHP. When the wave of depolarization gets inside the T-tube, it's going to change the shape of this dihydropyridine, and as a result, it's going to change the shape of this ryanodine, and as a result, ryanodine opens. And the calcium is going to get released. As you can see, calcium is going to come out inside the cell. You know the rest of the story. Calcium is going to attach to the troponin C, and as a result, tropomyosin is going to move away and contraction is going to happen. As long as calcium is high inside the cell, we're going to have continuous contraction. What do we have to do to end this one? Well, there is a pump here. It doesn't show in this picture, but this pump is a calcium pump. Using energy brings this calcium back inside the terminal cisterna. This pump is called CERCA. CERCA is a calcium ATPase pump. This calcium inside the terminal cisterna is going to attach to a protein. It's called calcicostrin, and this calcicostrin is going to keep the calcium inside this terminal cisterna until it is released. Now, let's talk about how a muscle is going to get shortened. When the head of the myosin attached to actin, that's called contraction. Sometimes contraction happens without shortening. Sometimes contraction is going to cause shortening of the muscle. If shortening of the muscle happens, that shortening is a result of sliding thin filament over thick filament. Just like this picture. As you can see, when the thin filament is going to slide over thick filament, the H zone is going to get smaller. As you can see in this picture, the shortest length of sarcomere that we can achieve, it would be as long as your thick filament, or A band, because after that You have to bend thick filaments to be able to achieve shorter length, and that is impossible. We call this idea sliding theory because it explains how your muscle is going to get shorter in length. So to explore this one, let's go through this series of steps and explain how this sliding theory works. We're going to start from the time that the head of the myosin is attached to the actin. Calcium is available and the head of the myosin is going to attach to actin. That is called rigor. Realize this end of the thin filament, which shows by negative sign, is toward the center of the sarcomere, and this end that shows by plus is toward the Z-disc. As soon as the head of the myosin is attached to the actin, that is called rigor. This is a very short step. Remember, there are ATP inside the cell. This ATP is going to come and attach to the head of the myosin. As soon as the ATP is going to attach to the head of the head of the myosin is going to detach from the actin. Later, the head of the myosin is going to wrap around the ATP. That is going to change conformation of the head of the myosin, and the head of the myosin now is going to cock toward the plus sign, toward the Z-disc. Head of the myosin is going to break down ATP into ADP plus P. Later, the PI, or pyrophosphate, is going to get detached from the head of myosin. Now the head of the myosin is going to attach to the actin again, but this time to the actin closer to the positive sign, to the Z-disc. This attachment is called power stroke, or it's called force. After attachment, the ADP is going to get released. When the ATP is released, now the head of the myosin is going to pivot toward the minus and pull this thin filament toward the center. This is what you saw in the very beginning. You have to realize that we have thousands of this head of the myosin. Some they are in this position, some in this position, some in this one, and so on and so forth. I want you to think about this one like a tug and war. When you are trying to pull the rope, you don't think that everybody at the same time they're going to release the rope to grab a further point of the rope. Some people still are pulling it, as other people they are just releasing one hand to grab a closer point of the rope. Not every head of the myosin is going to detach from the actin to move toward the positive sign. Some they are still attached, some is going to detach and is going to attach to the plus end of the thin filament. I also have to draw your attention to the fact that if there is no ATP, the head of the myosin will not detach from this actin. When ATP is not available, the rigor would stay longer. That's the reason we call the muscle contraction in the dead body as a rigor mortis. Because when someone dies, ATP is not available anymore, so therefore the head of the myosin is going to stay attached to the actin. That's why it's called rigor mortis. The step in the picture A is very short because as soon as this attachment happens, we have ATP to release it and attach it to the another actin toward plus sign. Therefore, we say to stop contraction, we have to remove calcium. Adding ATP only is going to remove one head, while other heads can be in different stage of this cycle. But to relax a muscle, the only way is using calcium pump to remove calcium. Again, ATP only is going to remove one head of the myosin from actin, but is not going to cause relaxation. Every time the head of the myosin is going to cock and get closer to the plus sign, is going to move by 10 nanometer. The second thing I want you to know is contraction and relaxation both are active events, because to relax muscle, I also have to spend ATP to make the calcium pump function.